Good afternoon, everyone, and welcome to this lightning lunch session. Hopefully you've enjoyed some of these sessions that have already gone on, and you're looking forward to some more that are coming up. Today, I am joined by Stefano Mariani. Now, if you have watched any of these sessions before, you will have known that Stefano has already contributed to these lightning lunch sessions, and it was a really, really interesting session. If you would like to catch up and you're watching on demand in the future, you can watch both of these sessions from early next week. If you're not familiar with Zoom that we are using, if you are watching live, then you can actually contact us at the end of the session using the Q&A. But if you are watching on demand, you can contact us at outreach@ljmu.ac.uk. If you are joining us live, then we can't see or hear you, so sit back and relax. And if you do want to use the Q&A tab, you can also anonymize yourself if you do want to ask some questions. So this session will last approximately 30 minutes. I will pass you over to Stefano, who will be talking about studying animals without seeing them, the tiny fragments of DNA that gives us more information.
Thank you, Ben, and hello, everybody. Good afternoon, good morning, and of course, any time of the day or night, you're going to be able to watch this and listen to this. Very pleased to contribute once more to showcase another large component of my research portfolio and a slightly more obscure one. I will start sharing screen so that everybody can see my presentation. And so, yeah, I mean, this is something that could sound almost like a provoking, almost like a challenge, somehow also something that is maybe perceived by many, maybe as something annoying, because hang on one second. I want to listen to something that can persuade me and entice me and excite me about studying animals and preserving their ecosystems. And now you are telling us that you can study animals without seeing them. Well, I mean, give me a chance to persuade and give me a chance to show you that actually there is a lot of attraction and a lot of exciting things about this way of doing science. Now, I am not crazy. I have started studying biology because I was myself also in love with animals, and I love to observe animals in the natural environment. I love to preserve them. I love to do everything I can to make sure that this beautiful balance of ecosystems that we have in this unique planet are preserved in a way that they can continue to sustain our future, because obviously a well-functioning planet, and especially a well-functioning ocean, is key to our, survival over a long term on this planet. And even though obviously I was fascinated by observing animals in the sea, especially like these beautiful slinger sea breams that are found off the reefs in the eastern part of South Africa, I realized that there was something beautiful that was basically beginning to become mainstream when I was a student, and was DNA technologies. Obviously, I think that most of us know what DNA is. DNA is, we could define it as a building blocks of life. It's the blueprint of around which every organism is designed. Every living organism, with the exception of a few viruses, have DNA. And even those few exceptions have RNA, which is a very similar molecule. And this DNA is essentially organized in this helix. It is packed into chromosomes. These chromosomes are packed into a nucleus and inside cells. Ultimately, when we use modern technologies to unravel the information that there is in DNA, we have these four little bricks, the A, T, C, G, popular also in popular culture and a lot of movies that have talked about sequences of DNA. But essentially, this codes the how we are made and how we function. These sequences of DNA, without going into too much detail, can be obtained most typically from a process called isolation of genomic DNA. And that happens when we obtain, we get our hands on an organism It can be a fish, or it can be a cat, or it simply can be even ourselves. For example, when we swab our nostrils and mouth to try to find the DNA of potential pathogens like the COVID-19, well, that is extraction of environmental DNA. In this particular picture, we have perhaps a small fish that is being unfortunately subjected to a not particularly pleasant operation of having the fins clipped. And from that little tissue, the DNA can be isolated. And of course, we can then sequence small bits of that DNA until, for a few stretches of DNA that allow us, for example, to get information about that particular individual fish. But there is another way. And the other way, it's called environmental DNA, as opposed to genomic DNA. Environmental DNA is when we actually collect portions of an environment, a sample of a larger environment, such as a pond or a river, or as you can see in that picture, I am collecting a bottle of water from the sea. Of course, what I could do also is to collect some sediments, some dust. But the water is the most natural environment of marine animals. And in that water, there are a lot of pieces of DNA. The big difference with genomic DNA is that genomic DNA belongs to one particular main organism. And eDNA, environmental DNA, short as eDNA, contains the DNA potentially of all the possible organisms that were swimming through that water. So yes, bacteria and tiny protozoans, many small microalgae, but also fish, dolphins, whales. However, the incredible thing is that we can have all of that in a bottle of water, and we can then eventually go down to the process that, again, I'm not here to come up with very complicated presentation with details and technicalities, but there are ways where we can isolate in one little test tube all the DNA that we had filtered from a large mass of water. And current technologies allow us to amplify DNA pieces from all the possible, or nearly all the possible animals that were present in that water. This is because every organism eventually loses pieces of skin, or bleeds, or also produces poo and releases eggs, and eventually these tiny little pieces of cells eventually break down and they release the DNA. Bacteria do that as well. So eventually you end up with a sample that contains a whole community: shrimps, fish, starfish, birds that have flown above the water or have dived to collect some food. So this is very fascinating. Now, the other thing we want to understand is what happens to this DNA. Well, this DNA is very short-lived. DNA in the water, once it's released, it's rapidly dismembered, disgregated, consumed, degraded by sometimes simple radiation, UV light, temperature. The molecule will break down because it's no longer living. It's no longer inside an organism that can replicate and repair it. Also, these tiny little fragments are also sometimes used as food by tiny bacteria that actually feed on these molecules because they are very nutritious. They contain carbon, phosphorus, and nitrogen that are essential for building everything about our bodies. So the interesting thing that you can see from this graph is on the x-axis, you have hours, time, and on the y-axis, here to the left, you have the amount of eDNA copies that you find in the water. These were experiments done in separate tanks where we had small blennies, so small coastal fish, and the common shore crab. It doesn't matter which species you're looking at, or it doesn't matter whether you look at the left panels that refer to the winter and the right panel refer to the summer. What happens is that when you look at different types of water compositions, whether they were natural seawaters or it was artificial seawater, once you remove the animal from those tanks, the chamois or the crab, after say 24 hours, you can see here on the x-axis, after about 24 hours, the amount of DNA decreases dramatically. And you can say that after 3 or 4 days, there is basically nearly no DNA left at all. What does that mean? Well, that means that when we actually go out at sea and collect the water, isolate the DNA, and detect the DNA of, for example, the oceanic white-tip shark that you see in that picture, we can rest assured that in that particular area, within the last couple of days, there was an oceanic white-tip shark. And the reason why this is important is that means that the detection of the DNA is actually quite a recent detection. And so you can actually trust that you are detecting something that really was there very recently, as opposed to a very distant sign, distant signal from a distant past. The reason why I'm showing you this picture is that because I want to give you some examples of why doing this environmental DNA analysis is, useful and interesting. Well, sharks, first of all, are very useful as an example because they are not as common as other fish. Here you have a graph with a logarithmic scale to the y-axis. If obviously you're not familiar with the logarithmic scale, I'm sure that this could be also for your math teachers to have a workshop about the usefulness of using logarithms. But in any case, you can see that in white and purple, there are the amount of DNA sequences found that belong to sharks and rays. And in purple, you have the amount of DNA sequences that belong to bony fishes, all the other fish. And you can see in these two real samples from the Bahamas, the amount of fish are like in the thousands. And in the case of the sharks and rays, we're talking about tens. Now, in the aquarium, and this was a detection on an aquarium. In fact, this was sequences taken from the Blue Planet Aquarium in Ellesmere Port. Actually, there was pretty much the same amount of sharks and other fish DNA, but that is because it's an artificial setting. And the big sharks and rays are overstocked in an aquarium because obviously they attract the audience and the visitors. By the way, from next week, we can actually visit aquaria again. Why this issue of amount of rarity? Because obviously eDNA can be very sensitive, and a lot of these animals are also elusive. They don't want to be seen by humans. Plus, to collect sharks, often we have to kill a lot of fish, put the fish on a hook, and wait for the sharks to bite the hook. Sometimes the sharks are heavily distressed, and you have to constantly check if a shark was hooked. So it is actually very time-consuming and also expensive, and also potentially traumatogenic and dangerous for both the operator and the shark. So getting some water from the sea and getting the DNA, it definitely feels like something that it should be very attractive in this context. And I'll show you the result of a study. I am easy to find. So you can see here, you go on the LJMU website and you type Stefano Mariani, you will find me. But there are also several Twitter accounts that I use, the personal one, and also the more professional one about the lab, where you can find, you can get in touch. So if you actually see something that you're absolutely in love with, and it's the things that I'm going to show you, then of course I can share the original papers that were published that contain all the details about this research, although sometimes they are very, very technical. But again, maybe if you are approaching your A level and you want to, focus on biology or similar subjects, maybe you can work on some of these examples with your teachers. So this is New Caledonia, a very distant archipelagos in the Pacific Coral Triangle. And all these little dots represent different techniques of studying sharks. In green is the underwater visual senses. That is scuba divers swimming and observing what they see. In blue, you have the BRUVs, baited remote underwater surveys, visual surveys, and essentially there is a camera with a bait that attracts the animals, and the camera records. And then you have the red stars. The red stars are bottles of water. Now, when we compare these three methods, and let's remember that the green underwater visual sensors were records that were taken over a period of 20 years. The underwater cameras were taken over a period of 2 years, and the eDNA bottles were taken over a period of 2 months. Nevertheless, when you look at this Venn diagram, the Venn diagram is used to see to compare the overlap between different sets of observations. You can see that the BRUVs in blue, the UVC in green are highly overlapped, and the eDNA is able to identify 6 extra species of sharks that were never identified and detected with the other methods. That is quite a strong message, not only because its DNA seems to outperforms the other methods, but also when you look at these graphs below, this is the sample size. Because if you look at the x-axis in green, the scuba diver surveys had almost 3,000 observations over 20 years. The baited cameras have almost 400 over 2 years. And the eDNA, it was a bunch of people going for a few weeks over there and collecting 22 bottles of water. So imagine how much less effort it takes to obtain more sharks detection. This is incredible. Plus, 91% of the water bottles, which means kind of 19 of the bottles that were taken, contain some shark reads, whereas only just over half of the videos recorded contained sharks passing by, and just only 15% of the scuba divers' data contained sharks detections. And that is true for very pristine areas and very impacted areas. I'll show you another thing, because this is pretty much the same thing, but it refers to Mediterranean fish assemblages. It's not sharks, it's all other possible fish. Again, you have these colors. In green, this time, you have eDNA. In blue and yellow, you have the underwater visual sensors and the baited cameras. And in red is the commercial fishermen. You can clearly see that eDNA, on the x-axis, you have number of samples taken, and on the y-axis, you have the number of species detected. You can see how eDNA, after a few samples, 60 or 70 samples, already reaches a very, very high number of species, over 80 species. The commercial fishermen collect even more species because obviously they have large boats and they fish day and night, and they also have more observations. But for the same number of observation, eDNA is you can see in this zoomed in set, if you compare, if you stick to maybe 50, then eDNA is the best at detecting species diversity. And not only species diversity, but it also gets a broader, fairer representation of the types of fish, not just the specific species, but also the type of behavioral traits. We call them in ecology functional traits. And I'm talking about fish that school, fish that hide under rocks, fish that are grazers, fish that are predators, fish that are nocturnal, fish that are diurnal. So all the different ecological adaptations are captured by eDNA in a fairer way. For example, you can see that the visual observations are very, very different from the fishermen observations, because obviously fish go out for deeper water, larger fish, and the observation with the cameras and the divers generally tend to have observations that are predominantly skewed towards fish that are very curious and not particularly bothered by the presence of humans, whereas the eDNA in green cover a more overall a more comprehensive distribution of all the possible fish type. Imagine you are on a theater stage and you have different actors doing different things. You have a killer, you have the detective that try to identify the killer, you have the kids, you have a teacher, you have a firefighter. Now, imagine a method that is only able to identify the firefighter and the policeman, and another method that is able to bias towards the teacher and the nurse. Well, eDNA, it's a bit like the one that identifies all the actors on the stage of the ecosystem. Another interesting thing, this is another study by a Danish group that showed the correlation of eDNA samples, collecting bottles of waters, of the presence of mackerel tuna DNA on the x-axis here, and the presence of whale shark on the y-axis. So why are they correlated? So the more of one you find, the more you find of one fish, the more you find the other one. And this is exciting because we know that whale shark chases the mackerel tuna, not because they eat them, because they're too big, but because they know when they spawn. And when the mackerel tuna spawn, they produce an enormous amount of eggs. And the whale shark go there with their large mouth and gulp all the eggs. So they know when they can find a lot of food. So imagine, instead of tracking this massive fish across the ocean, you can just go in an area over a period of different weeks and collect water. And when you find a lot of DNA from these two guys, you know that the mackerel tuna are coming to spawn, and the whale shark is probably around to eat the eggs. So this is not just about counting species, but it's also about learning from the ecological relationship between species at a much faster pace and lower cost than it normally is we used to do as marine biologists, as traditional marine biologists. What is the next frontier? And in the last few minutes, I want to tell you about something quite quirky that I've started recently, working on more passionately. And as you can see, these two guys are Marine Cusa and Chris Brody. Chris Brody is the one to the right, Marine is the one to the left. They are two PhD students of mine working at LJMU, and they are working on a fishing on a scientific vessel from CIFAS, the Center of Ecology and Fishery Aquatic Scientists. And they went on this vessel collecting these water samples. You can see in large parts of the English Channel, the Celtic Sea, the Irish Sea, the Bristol Channel, to study the environmental DNA in those areas. And it's quite high tech, right? Fortunately, this is a very well-equipped research vessel, but it's also incredibly expensive to run these surveys. And yes, if you have the resources, you can do these things, and you can have more resources. For example, the British Antarctic Survey is using a sea glider to collect samples, and potentially this could become a method to collect also eDNA. And there is a very expensive robot that was first pioneered in California at the Monterey Bay Aquarium, and now optimized at the Danish Technical Universities, also has sensors inside that are able to read the DNA of some species. However, the thing is $1 million. It's very expensive to deploy and to recollect. You need a satellite connection to transfer the data. So these are very expensive things, and they are hardly applicable and hardly usable in many parts of the oceans where we actually need to collect information quickly at an affordable cost. So once we were studying as part of the PhD thesis of a former PhD graduate of mine, Anjin Siegenthaler, the diet of the brown shrimp, which is a very common shrimp all around the British coast. This brown shrimp, obviously, it's tiny, and it eats, everything it finds around, more or less. And when you actually open the shrimp inside, and it's very difficult to see visually what they have eaten. So actually, DNA is the perfect way, because you just take the content inside the guts, you extract the DNA, and then you do all this sequencing to understand what critters are there. Now, there were all sorts of critters. There were worms, amphipods, isopods, different types of crustaceans, right? And there were algae, there were small mollusks, a lot of, hundreds of different species and tiny dwellers of the shallow coastal areas. But we also noticed there were fish. So obviously this tiny shrimp can only eat the fish when they are eggs or tiny larvae, or when the fish die and it becomes basically, a dead body, and they just go as scavengers and eat them. These are the fish that were collected in those areas with nets. You can see should go back. So in this dotted black line, and then this is the fish that has been collected with DNA from water samples, from sediment samples in orange, and from the stomach samples. Now you can see that there was a large proportion of the fish that were known to inhabit those estuaries, because these animals are very common in estuaries, could be found in the stomach of the shrimp. So the next question is, wow, we have a lot of organisms already present in the environment, plentiful, that collect by just eating what they find, they tell you what lives in there. So potentially you could have them as the natural sentinels, as the guys that do the job for you, essentially. But of course, nobody wants to kill too many shrimps, and perhaps the shrimp select certain things more than others. And then I started thinking, surely there must be something there in the water that it's even better for this purpose. And these guys are marine sponges. Think about it. They are almost in every ecosystem, marine ecosystem. They do not suffer pain. And if you take a piece of them, they regenerate. So, and they filter water. All they do, they filter water. Imagine a sponge the size of a football can filter as much water as you have in a swimming pool every day. So this constant filtration surely must pass through the body a lot of DNA that is present in that environment. So if I am able to collect a small piece of sponge, perhaps I can get a representation. Now, we are now working on a project that studies exactly that. And the reason why we are able to do this is that we happened to have some pieces of sponge that were in the freezer, that were collected for different purposes. And we decided to extract the DNA from them and try to look for fish DNA. And we had some of these sponges were coming from Antarctica, some of them were coming from Italy in the Mediterranean. And the samples were very different because we found the DNA of typically Mediterranean fish in the Mediterranean samples and Antarctic fish in the Antarctic samples. In the Antarctic samples, actually, we also found DNA belonging to seals and to penguins that were known to inhabit those areas. So this method works, and it's fascinating because there are potentially a large number of natural samplers all around the oceans of the world. We have jellyfish, we have sea squirts, we have sea anemones, we have mussels. And currently there is a PhD student of mine called Alice that is working at LJMU to use mussels. Yes, the mussels that you can see in the Albert Dock when you walk in town, it's full of mussels in the marina. Those mussels filter the water constantly. And perhaps if you take a few of them and extract the DNA from them, you just don't get the DNA from the mussels, but you potentially can get the DNA of all the larger animals that are swimming in those waters. And sometimes we don't think about that. So I leave you with just a quirky picture. The quirky picture is a lanternfish from an Antarctic project that is a very, very plentiful fish, and is in fact considered this lanternfish and considered one of the most abundant fish in the entire ocean. And we were studying the diet of this fish, and we found the DNA of the elephant seal. Now, obviously, these tiny little lanternfish are not able to eat the elephant seal, but they were picking some food in that vast Antarctic polar cold ocean. And as they picked some food, they were picking something in the particles that were present. Perhaps it was some pieces of skin or pieces of poo of 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 this vast, large, endangered animal that is found in those cold seas. So the message here is there: we can use DNA to study what animals eat, and we can also find in their diet precious, exciting information about what lives in their habitat. And I hope I have convinced you about all the possible, exciting avenues that stem from this way of looking at the water that surrounds us. Thank you so much for your attention. Thank you for a lot of the members of the team that I work with, and the funding bodies, and the institution, John Moores, that is really supportive of this type of activities. And also just remembering when I was in Belize, because the picture that I opened with was from a beautiful coral reef in Belize, and this is the research team in Belize. Most of the guys are from the United States here, but we're looking forward to the moment where we actually can go out and collaborate with people for real, and going in the field and collect more biodiversity information. Thank you so much.
Thank you very, very much, Stefano. That was absolutely fascinating once again. There is now an opportunity to ask a question if you do have one. If you are watching on demand, then you can, of course, get in contact with us via a different method. And that is either contacting outreach@ljmu.ac.uk or courses@ljmu.ac.uk. You can find more information on the Lightning Lunch talks that we have going on. There's a few more going on over the next 2 weeks. If you're interested in a variety of different subjects, you can find those on our undergraduate outreach pages under events. And if you are interested in any of the topics that Stefano has talked about today, I was chatting with Stefano before we started, and there's a couple of different modules that Stefano leads on across a couple of different courses. These are all within the Faculty of Science. And if you want to find out more about how you could potentially get onto those courses, you can email scienceadmissions@ljmu.ac.uk. But from both of us today, and to Stefano again, another fantastic talk, we will say goodbye. So bye-bye, everyone.